Hi guys, Tam here. Welcome to today's lesson. In today's lesson, we're going to be reviewing a little bit from last lesson on transcription translation, and we'll also be looking at post-translation modifications. So we'll come on to that in a minute. For your starter, if you could pause the video, and I'd like you to jot down the definitions of these key terms. You might want to sketch out a diagram as well, just to remind yourself what it looks like. So pause the video. Okay, and next I'd like you to pause the video and have a go at these two questions here. Okay, so what are introns and exons? Now, basically, introns and exons are what you'd find within a gene. So here we can see in this diagram, we've got a gene, and the gene is comprised of exon 1, then intron, then exon 2, then intron, then exon 3. So that gene is ultimately going to code for a polypeptide that can be used to make a protein. So our questions are where/when in the process of transcription are introns removed? Now, introns are actually removed after transcription. So here we can see initially transcription is going to copy and make an RNA copy of the whole gene, and that includes the exons and the introns. And then the introns are going to be removed after that RNA has been made to make a mature messenger RNA molecule. Okay, and so here we can see that the exon 1, exon 2, exon 3, and we've had the introns removed. So generally, when you're talking about these, think about exons as coding regions and introns as non-coding regions. So exons will be used to code for amino acids in the polypeptide chain, whereas introns, these are not used to code for amino acids in the polypeptide chain. And how are the exons joined or spliced? So here you can see they're actually stuck together or spliced together to make, again, that mature RNA molecule. So these are your lesson objectives. I'd like you to feel confident that you can describe in terms of exons and introns RNA synthesis, explain the basis of the one gene, one polypeptide hypothesis, and then also any further modifications and combinations of polypeptides in post-translational modification. Okay, so please can you pause the video again and try these few questions on here. So pause the video now. All right, and we're going to talk through some of these. So first of all, what implications does this hypothesis give for understanding how DNA protein synthesis works? So the hypothesis is called one gene, one polypeptide, and that implies that from each gene you're going to get a polypeptide made. And as we know, polypeptides can be used to then synthesize proteins. So here's our second question. Why do you think the hypothesis was changed from one gene, one protein to one gene, one polypeptide? Now we know from doing proteins earlier in the year that proteins can be made up of more than one polypeptide. So for example, hemoglobin is made up of 4 polypeptide chains. So in actual fact, each of these genes, as you can see down here in this diagram, each gene is going to code ultimately for one polypeptide. And then it might be that that polypeptide makes a final protein, or it might be that it has to combine with another polypeptide to make a fully formed functioning protein. Okay, so here you can see in this diagram at the bottom, in purple you've got one polypeptide, but maybe actually to make a functioning polypeptide, a functioning protein, we have to add another polypeptide like this one. So that's why it's called one gene, one polypeptide rather than one gene, one protein. And then finally, using the diagram below, suggest how post-transcriptional modification can produce different types of polypeptides from the same gene complex. So again, transcription, remember, this is what takes place in the nucleus, and this is where a messenger RNA molecule is made. So post-transcriptional modification is where we get this mature messenger RNA where you've had the introns removed, and we might be able to form different polypeptides from this depending on the order that we stack these exons. So you actually have a very cool molecule called a spliceosome. So imagine this is my spliceosome, for example, and the spliceosome could either splice together the exons in this order, so 1, 2, 3, or maybe the spliceosome could combine them in a different order, like 2, 1, and 3. Okay, and that's going to produce a different polypeptide, because now if our exon 2 comes first, you're going to get a different amino acid sequence initially compared to if you would have exon 1. So that would produce a different polypeptide. Please can you pause the video and try this activity here. There's a nice little video link with this as well that will just remind you of how those organelles work together. Okay, there's a little bit of a difference in the video, so you might notice them talking about adding carbohydrates in the rough endoplasmic reticulum, but just remember, for us at A level, we talk about adding carbohydrates in the Golgi body. And there's a few keywords as well that you might want to use here, so pause the video and try that. Okay, so let's have a look at the answers. Now, this is what we call protein trafficking. And here you can see we've got a diagram of a cell. And we know that in protein synthesis, we have the DNA in the nucleus. So here's my DNA strand, my chromosome. And we're going to copy part of that chromosome, a gene, and we're going to make an RNA, a messenger RNA molecule, and that messenger RNA molecule will move out of the nuclear pore to a ribosome. And then on this ribosome, this is where we're going to have a polypeptide chain synthesized. So here's my polypeptide chain being synthesized, and that polypeptide chain, as it moves through the rough ER, is going to fold into its three-dimensional specific shape. So here you can see as it's moving through, it's actually going to fold into its specific three-dimensional shape until it moves all the way through that rough ER and maybe ends up at the bottom, and here then it can bud off in a little vesicle, and that vesicle can carry the folded polypeptide to the Golgi body. And in the Golgi body, this is where we're going to have modification and packaging of our protein. So we might modify our polypeptide by adding another polypeptide chain, and or maybe another 3, for example, if we're making something like hemoglobin. We might add something like a prosthetic group while we're in the Golgi body, or carbohydrate chain to turn it into a functioning protein like an antigen. And then eventually, as that protein is moved through the Golgi body, it'll bud off again in another vesicle, and here you can see it's being carried to the cell surface membrane for exocytosis, and you can see here we've got the protein being expelled. And again, so this could be something like a hormone being expelled from the cell, or if it was incorporated into the cell surface membrane, maybe it would be something like an antigen. And this is protein trafficking. So this is how the protein is synthesized and sort of trafficked and moved through that cell. We're going to take just a few minutes to kind of fall in love again with biology. So if you could watch these couple of videos, there's just some really cool animations here on fireflies, and then we're going to actually do a little bit of research afterwards. So pause the video and go and watch those. And next, I'd like you to pause the video and take 30 minutes to complete ideally 2 of these activities, but obviously if you're working super quick, then complete more of them. But they're all about genetics. So do feel free to kind of explore this website and have a look at other links. You know, this is your time to really explore the science behind this stuff. Okay, so 30 minutes and then come back. There's a quick Quizlet, so you might want to do that as your plenary. And then just to review, hopefully you should feel a little bit more confident that you understand what exons and introns are, and how they can be, the introns can be removed in post-transcriptional modification. Then we should also understand about the one gene, one polypeptide hypothesis, and then also about further modification in the Golgi body, for example, and protein trafficking. Have a lovely day. Okay, bye.